15 years of work culminated in this super SOX protein, which increased reprogramming efficiency by like almost 50x. So like really exciting work, but it took 15 years. Ideally, in order for us to solve aging in our lifetime, we kind of need something that's faster than that. So I guess we were excited that we were partially like supersede those results in a couple months of work using the model. To actually getting to something like longevity escape velocity is to putting a model into an environment where I can actually just test hypotheses at scale. I am in the camp of people who believe that GLP-1s are probably going to show increased health span and potentially lifespan for all people.
Do you think abstinence increases lifespan?
Interestingly, yes. In aged cells, you try to reprogram them, you mostly see cell death. So there's a somewhat conspiratory sort of like question of like how much is reprogramming rejuvenation versus selection?
Welcome to the Free Radicals podcast, where we interview the scientists and builders working to dramatically extend human lifespan and bring about a sci-fi future where humanity has full control over biology. Today's guest is Rico Meinl, the head of applied AI at Retro Biosciences. Retro was seeded with $180 million by OpenAI CEO Sam Altman to develop therapies to prevent and reverse age-related disease. And it is widely recognized as one of the leading AI for longevity companies. In this episode, we discuss Retro's work with OpenAI to engineer 50x more effective Yamanaka factors, what it means to build foundation models that can reason across natural language and protein sequences, and why the bottleneck in biology is more experimental than computational. We also get into the biology of aging and how AI can enable therapies that dramatically advance healthy lifespan. I am your host, Daniel Schur, and my co-host is Eric Dye. I hope you enjoy this episode of the Free Radicals podcast.
Rico Meinl, thank you for joining us on the podcast.
Thanks for having me.
So I think, we have a bunch of exciting things we want to talk about with AI applied to biology. Let's first start out talking about Retro's partnership with OpenAI and the discovery of novel Yamanaka factors that were an astounding 50x more efficient, according to the report.
So essentially we took a existing GPT-4o language model. Okay, well, to take a step back, why would we want to increase the efficiency of Yamanaka factor reprogramming? So like, I guess one of the sort of like founding premises of Retro was the idea that you can take any cell type in your body and reprogram it to a pluripotent stem cell. And then if you were to redifferentiate it back to any other cell type, because a stem cell can make any cell type in your body effectively, you have a rejuvenated cell. So obviously you don't want to create iPSCs in a person's body because, that would have a risk of creating cancers, but it turns out you can go only halfway and still erase a lot of the aging marks of cell types in your body. And that would potentially make a pretty futuristic therapy because you could just rejuvenate cells in your body as well as ex vivo. The little problem with reprogramming is that it's extremely inefficient. Less than 0.01% of cells are actually reprogrammed when you drop these 4 proteins called the Yamanaka factors on them. And that efficiency further drops down in aged or diseased donors, which are obviously like the major target population for such a rejuvenation therapy. So I guess we were asking ourselves or I guess a long-standing problem that we sort of like were wondering about in the company is like, how would you go about increasing the efficiency? And people would change the media conditions of their reprogramming cocktail, or maybe they would change from viral delivery to mRNA delivery or something. But given that you have 4 proteins that are reprogramming, they're essentially the main drivers of this process, why wouldn't you just change the protein sequences directly? Protein engineering, obviously a thing, we can talk more about it, but it's sometimes often it's a very laborious process and not very straightforward, especially if the proteins are not very well understood. So we decided to tackle that with AI, and we had a partnership with OpenAI, and we decided to basically focus on this problem, take a model, make it a good protein engineering model, and apply it to this use case. And this is where essentially we ended up with this discovery.
And so can you tell us how does that model work? And maybe even walk me through what your workflow was like running it? Because to me, I don't know, there's a lot of different protein design models I've heard about, and they're very abstract. It's hard for me to understand
Right.
what they really are.
Yeah. So, okay, how do you characterize it? So there are protein language models. They are sort of purely trained on amino acid sequences, like ESM, ProGen, I think are the 2 major workhorses. There's of course structure models like AlphaFold. You give it a protein sequence, it will predict the structure. Now they also model DNA, RNA, etc. And And you have, of course, like models like GPT who have read like basically all the literature out there. You can ask them questions about certain papers and they'll be able to answer it, but they wouldn't do a very good job with protein design. So we essentially wanted to like try something in the middle where you start off with a GPT-4o language model. It basically already knows about the whole literature around reprogramming. And then you sort of like further train it on a bunch of raw biological sequence and structure data. So now you have a model that you can prompt with natural language, but it's also very good at designing proteins. So for example, if various ways you could prompt it, you could just literally ask it to generate a protein sequence for you. You could sort of like give it the first half of a protein sequence and have it generate the rest because that's what it learned to do during training. You could give it a protein and say like, I'm going to remove this core domain and ask you to refill that domain. So now you can only generate like one domain of a protein. You can prompt it on protein interactions. You can prompt it on like evolutionarily related sequences. So like where you show it, for example, a protein from a sheep, the same protein in the pig genome, and the same protein in a human form. And based on that, it would generate like whatever things would come next in that sequence. So there's various ways to prompt the model.
And so we have a ChatGPT model that was trained on like all of human language, right? Like the internet and books and all this. So it's a bunch of language data. And then you've added on another form of data that is trained on, which is like amino acid sequences and structure.
Yeah.
And so you're able to then, because of that, because it's a multimodal model, you can just speak with it the same way you would speak with ChatGPT,
Yes.
but it also speaks the language of the protein. So you can say, hey, this is my problem I'm trying to solve. Give me a protein sequence that will solve it.
Very much.
It can do that.
Yes.
There's a problem there of annotation, right? You're actually trying to translate between this language of amino acid sequences and human natural language, right? And so how do you deal with that problem of annotating and kind of translating efficiently?
I guess like it's all about data set composition, how you structure the data set that you feed into the model. I think LLMs sort of show that whatever data you give the models, they'll be able to learn because now we have general learning methods with transformers. So however you're planning to use the model, you structure the data in the same way and the model will pick up those patterns. So I think it was like just a lot of effort on how do you actually structure such a data set to enable the model to learn like patterns across these different languages.
So how do I think about, like, you could have models that are trained just on like biological data, amino acid sequences. You could have models that are trained just on language. I assume bringing them together doesn't just give you a nice user interface for a protein design model. Like, I assume it's also getting the advantages, right, of, something that's on the way to superintelligence.
As in the like so you mean essentially like because a normal GPT model, like I guess if a model were to truly understand the language of biology, it would be a superintelligence because humans are unable to understand the language of biology.
Sorry, that's an interesting point, but not what I was asking.
Okay.
What I'm asking is, like, what do you get when you combine a protein design model and an LLM? I'm guessing it's not just a user interface for the protein design model. I'm guessing because the LLM is so advanced and has so much data that it also gives you a smarter protein design model.
Presumably. I mean, I should mention like this is I guess not like in order to get like an LLM that you can actually properly interface with, you have to do all of these like post-training steps on top of it so you can actually chat with it. I guess like we're sort of like focused more on like the former part where like you remember GPT-3, like you still had to be very clever about prompting it. Like in order for it to become ChatGPT, you have to put a bunch of reinforcement learning on top of it so you can actually interact with it as a human. And GPT-3 could theoretically do basically the same things as like ChatGPT, but you'd have to be more creative in how you prompt it. I think we're probably at that like GPT-3 style prompting at this point, where it's like it's not yet a polished product where you could just go and like speak to it and be like, hey, make me that like new KLF sequence as like 50x better at reprogramming. You still have to be creative about how you prompt it. But like if you do the prompting right, you can get, I guess, the results that we were able to achieve.
I see. The other thing you're getting, I assume, is you're getting biological knowledge that exists not only in the form of protein sequences, because it's, GPT is trained on a bunch of science papers.
Right.
So that's the other thing. It's bringing a bunch of like research knowledge.
Yeah, for sure. Because like I guess for the task of reprogramming, you already have like a large body of like 20 years of literature. So like you could of course try to learn that all from scratch, but why would you, right? Like you can just use it as a prior to just build on top of it. Because I guess I think that's what you really care about with these models, right? You don't just want it to regurgitate existing knowledge. That's useful for human researchers, but then really you want to build on top of it. So why not just build on top of the existing corpus of knowledge that we already accumulated?
When you're engaging with models like those you're building in-house or other models created by other parties, what is the layer of natural language that you're using to interact with these models? And what I mean by that is like we can imagine there's different kinds of semantic layers that would make sense to engage with a protein language model, right? There's like the specific language of like I want a protein binder that hits this very specific set of epitopic like residues, right, that I want to bind. Or you could go a layer above that and say I want to hit this general protein and have this sort of function. Or you can even go a layer above that and say like I want to cure this disease, right? And so what layer are we at right now in terms of like semantic engagement, or do all of them work in some capacity?
Interesting. I always think that I think what's like becoming more and more possible is that you have a mechanism, you kind of know that it works, reprogramming, for example, and you want to improve upon it. I think models are pretty good at that, especially because you can like have your model design a bunch of sequences, you can test them in a lab, and you can use that information to feed it back into the model to get like a better next iteration. I don't know if we're quite at the point yet where, and those would be like truly fundamental breakthroughs, if you could go from 0 to 1. I don't know, like there's like what people call the delivery problem, right? Like it's very much non-trivial right now to deliver stuff into cells safely in the body. There aren't really any great solutions for it, except just like mimicking what a virus would do, for example. I don't know if a model would be able to go currently in the current form from like 0 to 1, where it would just tell you, here's what you need to attach to your viral vector, for example, and then it would just like instantly deliver a great fraction of human cells instead of like a couple percent that we get right now. Like so I think we're very much in the regime where like you have a problem, you have like a somewhat working solution, you want to improve upon it. Like maybe you have like a very weak binder and you want to get a strong binder. I think that's probably the regime where we are right now. But it would be very interesting to go one level above that.
Right. And so the way that we would like take this back into the actual space of like I'm working with a model to get a stronger binder against a known target, you would say, look, I have these existing binders with this sort of experimental, like K off, K on, values, and now I'm looking to get something with a greater KD, right, or lower KD, whatever it is, right, binding efficiency. So can you just generate new designs for me based off these known designs that potentially have a greater binding efficiency? Is that
as an example? Yeah.
So could you walk us through the process of like this model that you created in this partnership with OpenAI? Can you walk us through the process of training that model?
Well, I guess you kind of think, well, okay, so one thing I do want to point out is like, and this has been getting like somewhat mixed up in some of the publications. Like this is not a specific longevity model, nor a like Yamanaka factor
We're
model.
not interested in any more then.
Right. Well, I mean, it's a general model, and we've applied this to the use case of improving the Yamanaka factors. We did have actually other use cases in house where we also got like results that we haven't published yet. But it's just like underlines like this is a general model. It can generally be used to like re-engineer proteins. Yamanaka factor was a use case that we found very interesting, was very relevant for Retro. We published about it. I guess how do you approach it? Like, what does a model need to be able to work with proteins? Like what kind of design tasks might you have? How would you want to prompt the model? And I think from the question of like how would you go about prompting it, you want to work your way back to like what data set do you need? Protein sequences are the first thing that's like incredibly useful, and we have a ton of data there on the internet, like literally billions of sequences. Why are protein sequences useful? Because the Yamanaka factors, for instance, are just not very well structured. So you put them into AlphaFold, and you basically get a tiny structured domain, and then just like spaghetti arms around that structured domain. And it's just like not that that region actually has no structure. It actually does have structure in the context of the cell upon binding certain partners. It's just like we just don't know about it because these domains are really hard to crystallize. So there's no data in the PDB. Therefore, AlphaFold doesn't know how to model them. So for those proteins specifically, you actually don't like the structures is not that useful. Like you want to work based on the protein sequence because there's like a little language of like interactions encoded in the sequence itself. So protein sequences very useful, lots of data. Protein structure. I mean, you have both like the PDB as well as like bunch of distilled data, obviously, from various sources. Protein interactions, both on the sequence and structure level, like coevolution, basically like MSAs, like these multiple sequence alignments where like, you can infer, like I guess AlphaFold, for example, like took a protein sequence and it runs like a query against a massive database of other sequences and like finds similar sequences. And then that would be the input for the model, and it would be able to pick up a lot of coevolution in the input and then create your structure from that. ESM has then shown that like you can train a protein language model, and then your model can go from a single sequence directly to the structure, and it basically matches AlphaFold in performance. But there's still a lot of usefulness in having those multiple sequences and giving the model the whole context of across evolution. So that would be something you can add to the model.
Just on that point, my understanding of ESM's fundamental update from the work done on AlphaFolds, and this is from like a couple years back, so I may have like forgotten some of the details, but essentially the ESM algorithm was MSA independent, right? You didn't need MSA in order to recapitulate a lot of the results of AlphaFold, whereas AlphaFold is like very dependent on MSA.
Correct. ESM would implicitly learn the MSA by just learning on protein sequences.
Remind the people who don't know, not me, MSA and ESM.
Yeah.
MSA, multiple sequence alignment. So this is the process where you take a single sequence, you query it among a huge database of sequences, and you basically get a sequence alignment. So now you can, for example, see, oh, this domain has been around in that protein since like microbes, and since then it's just like the protein has added like other domains to it, but that core domain is very well conserved. Like that's what an MSA would tell you.
And you can infer that for whatever function the protein is serving, like that domain is very important for it.
Very much, yeah. Because that's why nature kept it around, right? It's like the that like photo from the planes that came back from World War II, and they have,
Oh yeah, the little dots. And
that's what an MSA is, right? Like
Yeah.
like everything that you don't see anymore probably was detrimental. And then ESM is like, what is it short for? Evolutionary sequence modeling.
Evolutionary scale.
Evolutionary, yeah.
Yeah.
Evolutionary scale modeling. It's like, I think probably the first like really successful mass language model that showed that because like I guess now we have GPU acceleration of like multiple sequence alignment, and it's like not that big of a bottleneck anymore. But when AlphaFold first came out, like these are pretty heavy computations. Like you would maybe wait for like half an hour for it to like compute your multiple sequence alignment, whereas like if you train a very large model on just individual protein sequences, and because it has seen all the individual protein sequences, now it has basically implicitly learned the multiple sequence alignment for any given sequence. You can essentially use it as a dynamic database for your protein alignments. So it's effectively like ChatGPT is effectively like a compressed version of the internet in a language model. And ESM is a compressed version of all the sequences, all the protein sequences in a protein language model. And then when you map from that compressed representation of sequences to structure, it's just like at that point, it will have already learned all the coevolution and structure domains that are necessary. And then you just map it to the actual 3D structure. But at that point, it becomes like a relatively trivial mapping problem.
So that helps me understand like how the model understands the relationship between sequence and structure. What I don't follow is how you get from structure to function. Like, where is the model given that information?
It's not. I mean,
Well, that clears it up.
Good question. Yeah.
I mean, there's like the I think people had this assumption that like structure is function, and like in a way, it is, right? Like, I don't work a lot with structured proteins, but like I guess like a receptor is a great example, right? Like you have a structured receptor, it has a certain like domain outside the cell, and it could be recognized by certain binders to catalyze like a reaction that would either like inhibit the receptor or activate it. Maybe it would be internalized in the cell, and that like, I guess, quote unquote function of the receptor, at least that part of the function is encoded by its structure. So I think this is potentially where this is like coming from. So I do think there's some truth behind it. It's not, of course, like not all function is determined by structure, but a good amount.
Well, it's also just like, like the way that I think about this at least is that structural biology is kind of your determines the fate of how you interact with other protein partners and other
Right.
molecular partners in the context of a cell or in a biological system. And those interactions ultimately form the basis of what we call either like signaling cascades or molecular biology, or if you get to like really high level about this, like systems biology.
Right.
Right. And so like it's the function of any given protein is ultimately like the integrated interaction like networks that it engages with, and like the how those interaction networks then cause some sort of biological interesting functional output of that biological system. So it's a very vague definition, but one that's like kind of like always true, right? But yeah.
I don't, so I totally on board with like, function follows structure. But I guess like what I don't understand is how are the models given any sort of, I guess, like structure dynamic data? Like how does it understand, like a biologist can look at one structure, another structure, and say like, oh yeah, I see how those would bind.
Mm-hmm.
How does the model understand that?
I don't think a biologist could figure that out, actually. I think
Really? But like,
it's
I mean,
impossible.
in my biochemistry class, right, they like walked us through the structure.
Totally fake. That's totally fake. That's made up. Well, I mean, I think Rico raised a good point, which is that there are these things called intrinsically disordered regions, which actually make up the vast majority of proteins, right? The protein regions. And that's why you look at things like sequence. You can't look at things strictly like structure, because structure comes from these really basically artifices of like using X-ray crystallography or something like NMR or some other techniques that give us very particular instantiations of protein structure. But in reality, like most of a protein structure is very floppy. It's also highly dependent on its context of like the ions. It's surrounded by the temperature, the water molecules, other binding partners. And so we actually have no real way of measuring like the true structure of a protein. And so you can't look at a protein sequence and tell like this is actually the structure. You can get a very specific instantiation of the structure in a very specific context.
Dark protein.
Fair. I mean, that makes sense. So then how does a model tell you, oh, your objective is this, here's a protein that can do it? It just makes no sense to me that it can make that leap.
I will say that like mapping an arbitrary function to a protein I don't think it's a solved problem. Or taking a given protein and asking sort of like what is the function, like in terms of true extrapolation, where you show the model a protein it has never seen before, which is already very difficult, right? Like most LLMs and protein language models train on effectively all the data that's out there. So it's already pretty hard to find like something that's truly out of distribution. And then people are also like, why does it matter? Why does it need to be out of distribution? We just train on everything that you would possibly ever encounter. So you just never have anything out of distribution.
It's like a wishful thinking type answer, but yeah.
Right. So it's like, but yeah, like measuring true extrapolation, I don't know, like, I think it's a difficult problem.
So
ideally you can set up your data set in a way that your model never has to do like large extrapolation from the source data set.
But I guess to pick an example, like the model, like the model that you guys were working on, like it outputted a sequence that suited your objective of making more efficient Yamanaka factors. Like, how does the model do that? Is my question.
Well, okay, so there's a component of testing these sequences in the lab. So I guess it was not like we spent like 6 months prompting the model. It generated a sequence. We tested it in the lab, and it was the thing, and then we just published it. It was essentially like we ran these like 3 screens. So we started with a pilot, which was like to validate the wet lab platform. So we have a platform for reprogramming where we have like some skin and like connected tissue cells called fibroblasts. Everybody uses them in the reprogramming literature. So like these cells attach to the bottom of your well. You just have your well with your fibroblasts in there. then you use like, for example, lentivirus to like actually get your proteins into those cells, and it takes like 2 to 3 weeks until they are like no longer fibroblasts, and now they're like, instead of being these like elongated flat cells, they're now like round happy iPSCs cuddled up in a colony, and they have like various markers that you could test for. So that's like your screening platform. So in order to make sure that it's actually working, we had like human scientists design like new versions of like SOX2, for example, to see like how, like, what's a human baseline for actually being able to like improve upon the existing Yamanaka factors. They did well, not super well. So then we had the model basically take in some of that information, as well as just like prompting it from just like base information of just like how these sequences look like across evolution, and had it generate a bunch of retro SOX sequences. Those did better than the human generated ones. Still not anything close to like 50x. Maybe there was like a 5x in there or something. And then we basically like generated retro KLF sequences. And this is where we got like some pretty exciting results before Christmas, where like suddenly the markers that we got jumped up from like, maybe like 2% to like 16%. And we're like, whoa, okay, something in that pool of things that we added to the cells must be working really well. So we took those things and like further validated them. And like obviously all that validation took a lot of time, and it took like a couple months until we were able to say like, okay, we now took these variants to like multiple challenges to like shoot this down, like shoot the hypothesis down that they're actually that much better, and we just failed to do so. So we were left with like, seems like the model actually generates some really good sequences.
So in this case, the objective function is like, I'm probably going to butcher some of this, but the objective function is kind of twofold. The first objective function is I want to get a diverse set of sequences that look like but are different than the training set here of like existing KLF or SOX2 sequences from like the entire library of all sequences we know, right? And the second is I want to improve upon this like programming efficiency measurement. And if as long as I'm operating within those two spaces and I'm improving on particularly the second objective function, then like we're moving in the right direction essentially, right?
Yeah.
Right. I guess then in that case, your objective function is the efficiency of reprogramming, which means like you've now introduced some type of like function data that is being correlated with structure and sequence.
Not structure, just sequence.
Just sequence.
Just sequence. We don't know the structure
right.
of any of these. We indeed, no structure.
Theoretically, we could have tied it in with structure, but in that case, we mostly did it with sequence. As I mentioned, like the proteins are not very structured, so.
Okay.
But yeah, like
seems reasonable. I believe it happened. I believe it.
So yeah, I mean, you're implicitly telling the model like what worked well by like showing it some of the examples of stuff that worked well, and it can use that information to generate like the next iteration.
Okay.
You could also do like a sort of like lap in the loop approach where you actually do some like reinforcement and fine tuning on top of it, but you don't really get to that.
Yeah, but if I'm picturing, like, if I replace AI with human, I'm picturing, like, a human basically looked at, like, oh, here are a bunch of experiments with a bunch of sequences. These were the results. What seems to be in common with the ones that had good results? What's different from the ones that had bad results?
Mm-hmm.
Oh, it's like these domains seemed, like, more important or something. Like, it's just recognizing the same way that, tell me,
in theory, right?
That's, like, in theory what it's doing.
I mean, this is, okay, so, like, I guess our baseline was sort of these, so there's this work from, I think, 2 or 3 labs that, like, over the course of 15 years did, like, sort of like human protein engineering, I mean, just like protein engineering, not human protein engineering, of, like, these, like, SOX factors and OCT factors during reprogramming. And it was essentially, I mean, this is really, really cool work, and it was what inspired a lot of our, like, us even taking on this project. But it's, like, how people would like rationally design a protein, right? Like you look at the like OCT-SOX interaction in like structured form, because their structured domains do interact. So you can look at like the sort of like sites at the binding interface, and you could sort of like rationally manipulate them and then test if it improves your programming efficiency. And then you can also like do something like a directed evolution screen, where you would essentially take your SOX factor, you determine like 3 amino acid sites that you want to swap randomly into every possible other mutation. So now you have like 3 sites by 20 different mutations. So you have like 8,000 sequences. You can go test them in the lab and basically enrich for the cells that are most reprogrammed. So then that way you can screen, not model based, but like you just like essentially do like an artificial evolution in the dish to screen for better factors. So it did that, And then, so the rational approach, there's like directed evolution approach, and then lastly, you just like what you said, like you identify the domains in like related SOX proteins, and you just like try to swap them around and see if there's something there. Turns out that actually works really well for like SOX2 and SOX17. When you swap their domains, you can make a more efficient reprogramming factor. So like 15 years of work culminated in this super SOX protein, which increased reprogramming efficiency by like almost 50x, and also just makes your pluripotent stem cells more naive. So now you can actually like go and like reprogram certain animal species that previously were really hard to reprogram. So like really exciting work. But I guess I'm wondering about it from a perspective of like, it took 15 years. Ideally, in order for us to solve aging in our lifetime, we kind of need something that's faster than that. So I guess we were excited that we were like match and partially like supersede those results in a couple months of work using the model.
I think what's really interesting too is this there's like a bunch of different ways of designing search algorithms in biology, right? Like you mentioned a few of these, which is like rational modular engineering, right? You're like mixing and matching different known entities or known components and then combining them into like super SOX, for example. And there's also directed evolution. There's also kind of like language model based like search. And I'm curious, like, are there any other like novel ways of going about prosecuting like protein space search that you're really excited about, or that's like kind of emerging right now?
There's some really cool stuff happening in like I briefly engaged with the like binder literature, and there was this like a contest last year.
The adaptive one.
Yeah.
Yeah.
And the winning solution, the Bindcraft, I thought that was a neat idea to essentially do like an in silico screening approach with like an inverted AlphaFold model. I do think that could be potentially really interesting to have a way to use your model as an in silico oracle to like screen a bunch of things to then have to test less in the lab. Because I do think the lab is a bottleneck for everything, right? So like having quick feedback like is great, but you will still be talking about like days of iterations and not like hours. And ideally ML scientists always like hours of feedback iteration.
Yeah. That's interesting. So let's walk through that process a little bit. Essentially, what you're proposing is that the way Bindcraft works is that you're able to use like an AlphaFold or AlphaFold like model to do in silico testing of protein-protein interactions.
Yes.
And then from there, you can discard those protein-protein interactions which seem very unlikely to be like real protein-protein
Essentially,
interactions.
yes.
Yeah.
Yeah.
And then you only actually physically test those that seem more likely to be like energetically favorable.
Yeah, exactly. I think you're basically on point. I also like also back to your question. I think like with LLMs, I think the interesting like what people would do is they would use LLMs to do like mass language models, for example. There's some papers out there where like to increase the protein stability, you can use a model to like introduce these point mutations and then score the likelihood of that with like a model like ESM. And then there's also the approach that we took where essentially instead of like making conservative mutations to your protein, you just have the model generate whole new protein sequences and just like throw those in the lab and test it. And I think people are probably not doing enough of that because I think there's a lot of like pushback on like, well, I guess if you wanted to make this into a therapy, changing a lot of the sequence is actually not desired because of potential immunogenicity concerns. But I'm like, you can always just like do screening like that in like a larger space and then just like reduce the number of edits to get back to something that you could actually use in the therapy. So I do think like I don't know, that approach just opens up a lot more exciting possibilities to discover like truly new science.
Mm. I'm reminded a little bit as well of a paper from Eric Kelsick and some other scientists from George Church's lab from a few years ago. It got published in Science, but it was on kind of the search space of capsid design for adeno-associated viruses, so AAVs. And so the core of the paper is essentially like you have this however many like multi-thousand base pair long AAV genome, and you're essentially trying to determine like what is the language of the AAV genome, like amino acid protein structure function search space. And so they tiled individual base pair mutations throughout the entire AAV genome, and then from there measured using like a many against one screen where they like test like thousands or millions of these AAVs simultaneously in a mouse and then use single cell sequencing to see like where do these AAVs actually end up, right?
Right.
Sounds really, really cool in theory, but I think it's much harder in practice. But yeah,
You mean
like,
setting up the platform?
setting
Or?
up the platform, and then eventually like the idea is you can eventually combine multiple of these individual point mutations to have like a super AAV that's like enriched for brain targeting, or
Right,
has
right.
a larger capsid size or things like that, right? Like payload size that it can take on. And
Oh yeah, I mean that's such a fascinating. I actually, it's funny. I had a deep research conversation on that this morning. Because you have like multiple bottlenecks there, right? Like you have the tropism of the AAV that like targets certain cell types, and tissues. Then those are uptaken via receptor-mediated endocytosis, and then they're stuck in these endosomes. And then apparently, like their ability to get out of those endosomes is also cell type dependent.
yeah.
Because different cell types would have like different acidic environments in those little like endosome bubbles that would even allow the AAV to escape. And that's like a huge bottleneck for LNP delivery, for example, right? Because like only maybe like 1% of your LNPs are actually able to escape those endosomes. For AAV, potentially less of a problem, but it's such an interesting it's a very interesting like problem space for delivery in general. I do hope that certain ML solutions could help give us a leg up there because it would just enable so many new therapeutic approaches. Like I feel like talking to so many people is always like why wouldn't you do X? And it's like, oh, because you can't deliver it.
Something there's a long list of things here that I don't understand. Another another thing that I don't understand is when we're thinking like the models are trained on sequences, and I get like there are some models that have structured data, but it's pretty janky, right? Because like crystalline structure, whatever. Okay, but we have the sequences. Let's say it suggests some sequence for you to try in the lab.
Hmm.
When I think about how a cell makes a protein, there's a lot that needs to happen for it to take on the right tertiary structure, right? Like there's a whole process there, and it's so dependent on the environment.
Hmm.
So that's another thing here that like I just don't understand how the models can like account for that. Especially when you give it all the sequence data, like sequences can give you different structures and different
Right,
functions depending on what cell they were manufactured in.
right.
So
I
what's
mean,
up with that?
there's a ton of like also post-translational modifications that are like very like cell type or like organism dependent. Yeah. Um, I mean, this is another point, right? Like, it depends on. I guess like we delivered initially via like lentivirus because it's like at that point you're just like trying to run a screen, so like you're not trying to make your life unnecessarily hard. So you're going to use the best tools like for the job, and I feel like they're like you're cheating a little bit, but it generally seems like you're able to express your proteins and they go through like the standard like pathways that a protein would go get the standard post-translational modifications. And if not, because you maybe like there's a phosphorylation site and you swap that site to like an alanine or something, and now it can no longer be phosphorylated, maybe that's a feature. Maybe that will like lead to change function, and that's actually like the reason for why your proteins perform better.
Hmm.
Um, so I do think a lot of that manipulation can be very interesting from a function perspective.
I have this like these a lot of interesting anecdotes about exactly what you're describing, Daniel. But one example of this is apparently in like mouse studies. Like you can systematically determine that mice who exist on like the way that mice are stored is these like rows of cages, right? And so they
Right.
like are basically have like a wall full of cages from the bottom to the top, and they have like clear plastic that they like roam around in. And there's all these sorts of examples like the bedding that the mice live on really matters. And so if they live on wood chip bedding, the mice will eat like a bit of the wood chips every day, and it'll dramatically change their gut microbiome, right? And so like that's an example. It's like obviously changes all the results of whatever studies you do on them. And then the mice who are on the top level of cages are exposed to more of the top-down fluorescent lighting,
Mhm.
and so they sleep much worse and it vibrates a lot more, and so they're just like much less healthy, right? And so it's like there's all these examples of things where it's like you could there's too many variables to account for, but they fundamentally affect the nature of like the data you're getting. And obviously, like any models that you're training on top of that data are going to be like implicitly like affected by that bias. But how do you account for it? I don't know that you can really account for it, right?
There's always this moment when you're like, why does anything work at all?
Yeah, that's
my
I'm
reaction.
actually surprised anything
Yeah.
works, you know?
Yeah.
Yeah.
I mean, I generally feel like, I mean, this is why it's so important to have like really strong effect sizes, right? Like, because if you have weak effect sizes and you have all these confounders for like cell or mouse studies, like you just can't really believe anything. But that's such an interesting point.
So on that point then, what is the effect in general of effect sizes, yes, and also, like, amount of data,
Mhm.
right? Like, what is the effect of scaling these models in general?
Um,
And sorry, actually, how do you made this point of like, "Oh, well, we have everything in the distribution is in the model. It's seen every protein."
right.
So what does even scaling it mean? Have you reached the limit? That's, like, everything it can have?
Um, I guess scaling in bio is, like, different than scaling on the internet because the data distribution is just, like, kind of fundamentally different, right? On the internet, you have a ton of junk. Like, basically, like, 80% or more of the internet is just junk. So you need really good filtering processes to, like, take all that data and actually get to the real signal. Like, bio is much less junk because you go to these sequence databases that are already sort of pre-filtered. So, like, UniProt, for example, has, like, 300, 400 million protein sequences in there. Like, these all went through certain filtering processes where you can basically ensure that, like, there's like very little junk in there, but then you have a lot of, because of evolution. Like, if you look at a human version of SOX2 and a mouse version of SOX2, they're like, what, 95% similar? So, like, is your model really gonna learn that much from seeing the same sequence as, like, 90% similar, like, over and over again? So you have to do a ton of, like, deduplication and, like, filter, like, diversification of your data. But I guess, like, and it also depends on the use case. Like, I guess, I don't know, I can't really share much about our scaling law studies, but, like, a good example is ESM. When they scaled up the, like, size of the model for just protein sequence modeling, like, a staggering feature was that the protein, like, the implicit learning of, like, what the protein structure might look like, just, like, linear, like, basically increased with the scale of the model, to the point where maybe for, like, sequence level tasks, their 650 million parameter model is, like, the best because the bigger model is just overfit, so it won't actually give you useful predictions. But for structure prediction, the 15 billion parameter model is the best. But yeah, I guess, like, in bio, your model would saturate a lot sooner than for the internet, because you have, like, maybe billions of useful tokens, whereas on the internet you have trillions.
I guess the problem here is that right now for the vast majority of um uh you know the vast majority of training data that we have for biology is derived from natural sequences.
Mhm.
And those natural sequences have, a few different evolutionary branch points. For the most part, it's like there's a lot of deduplication that we need to do to get enough structural diversity where we're not just like training on the same thing over and over again. But I think what we haven't really hit yet is this point of like the breakaway data generation of like artificially generated proteins, right? And that will
Right.
start to give us a lot more diversity and
Yes.
hopefully like a lot more like useful, interesting spaces of biology that are previously unexplored to go tuck into, right?
Right. Yeah.
Yeah.
There's also these cool, like, this is becoming more and more sort of, like, popular, and there's companies that are, like, solely focused on that, like metagenomics, where you basically go, I mean, this is how drug discovery was done, like, I don't know, 30, 40 years ago, right? Like, you drive into, like, some jungle in the middle of nowhere, you scoop a bunch of poop, and you sequence for all the bacteria that are in there. So it's like metagenomic because you have a massive sample of, like, poop where there's, like, a ton of different, like, bacterial genomes in there. And then you basically map all your protein sequences. So now you have, like, a huge diversity of protein sequences that may all have very unique functions because those bacteria, or, like, whatever it's in there, lives in, like, the most extreme environments. And there you actually have a ton of diversity, like, sort of, like, for creating new proteins and having a very diverse data set. I think this is very interesting. Maybe for making, like, new medicines in the short term, potentially less interesting. Um, but yeah, if you want to, like, catalyze, like, some crazy new enzyme, like, maybe that's the kind of data you want to train on.
It's like base camp research.
Exactly. Yeah.
Maybe we should talk a little bit more about how all this relates to the frontier of longevity.
Yeah.
Like, what does that
Let's
look
do
like? Like, why
it.
why does this matter for anything we're doing in longevity?
Um, there's this cool paper in 2008. I forgot the name of the last author, but they showed that, like, the popular H1 gene, um, if you essentially take that protein and do a single mutation, you can increase a worm's lifespan by 2x. If they take that same protein and basically cut it in half, so you remove, like, two core domains in that protein, you extend the worm's lifespan by 9x. So these are, like, among all the model organisms, like, the most, I'm pretty sure, like, most significant lifespan enhancing results that we've seen, and it's effectively a form of protein engineering. So that, like, really begs the question, like, protein engineering is very powerful. Like, what if the means to increasing healthy human lifespan are just not within like our natural genome. And like maybe you need to re-engineer proteins to actually get larger effect sizes. And I feel like that's really what our like Yamanaka factor like result like signals to me, is that by re-engineering proteins you can drastically increase effect sizes. So like that result is great. It's like off to the clinic. It'll hopefully be useful for patients. But what else can you do with this? And that's what I'm really interested in.
That's super interesting. There's the space of naturally occurring proteins and transcription factors. And then, I mean, that's like, an infinitesimally small piece of the overall space of potential factors.
Right.
And I guess what you're saying is your bet is that the things that are going to give us radical life extension, radically improved health, is going to be in that huge open space of synthetic factors.
Yes.
And there was a long pause.
Yeah, there was a pause. Wait, explain the pause.
There's many ways to skin a cat, right? Like maybe it will end up being a great like place for discovering what are the right targets, but then maybe there's other ways of drugging those targets. Like maybe there's a certain protein mutations that like basically makes your protein like 2x more stable. But then you could get the same effect by using a small molecule to like 2x upregulate your protein expression, and maybe that's like a better drug in the short term. So I definitely think it will be an extremely useful discovery tool. And I do think in the long term, I don't know, I have this like thinking that like maybe like, humans have like these phases of like from 0 to 25 you're in maturation phase, then from 25 to like 50, 60 you're in like the stable phase where like nothing really changes. Like you slightly age a little bit, but unless you're unlucky, like you're in a very stable phase, and then it's like exponential decline. And I think the first 25 years are like a massive developmental filter on like why don't we have many like GWAS targets for like that we find in centenarians that are like variants that like lead to massively enhanced lifespan. I think development is a huge filter, very likely, for many things that will be beneficial later in life. They would hurt during development because like this very fine-tuned process between various like transcription and other developmental factors. But that doesn't mean that like it wouldn't be useful to take a, I guess I don't know, this is like not maybe like next 5 to 10 years, but eventually like you take a 25-year-old and just like directly engineer the proteins that we found to be useful like into people, so they have like a much more healthy aging process.
It's,
Go ahead.
I'm just reminded of one thing, which is this recent paper from, I think, Cell that we've been talking about for the past couple of weeks, which is this this gene FOXO3. And so FOXO3 was originally identified, I think, back in the 2000s from these super aging centenarian GWAS screens. And they looked at various countries, including, I think, Japan, Germany, some others. And they found pretty consistently that amongst these super agers, these people who are consistently reaching 95, 100 years old, well, you only get one chance to do it, but they were reaching this age, and basically they had an enrichment of certain base pair swaps, like single nucleotide polymorphisms, at the site of FOXO3. And so like this has been like a bit of like an ongoing saga for the past couple of decades. But this paper that got published the other week was really interesting because
this is the MSc paper, right?
This is the MSc paper, and so they basically engineered in a constitutively active version of FOXO3 by swapping out an alanine into some other I think a serine or something. And so that constitutively activates the FOXO3 in these human NPCs, and then they planted those into NHP, non-human primates NHPs, and saw like dramatic changes in biological, potentially like at least biological age through measurement of like DNA methylation clocks and some other functional measurements. So I think that's an example potentially of like these like GWAS screens actually converting into something that could look like, it's starting to look a little bit more like a therapeutic intervention, right?
For sure, yeah. I guess like that one, and then there's ApoE2, which I think also consistently popped up in some GWAS studies. ApoE4 consistently having detrimental effects. But I mean, like, why is it only like 2 or maybe like 3 that pop up consistently, right? Like, I do think there's this developmental or other filters. I guess one interesting thing is like ApoE, I think it's mostly active like in the blood. Like it's not an intracellular protein as far as I understand. FOXO3 is like a transcription factor, and it's active in like all tissues essentially, like maybe that's also an effect. Like a factor that is like very specifically active in like only a few tissues would just not come through in a GWAS study. But if we did like more tissue specific GWAS studies of centenarians, maybe there's more that we can find there.
It's interesting.
That's very interesting. One of the ways that you could get to that would be, I guess, through like RNA sequencing, because you would see which genes are active in each tissue type.
Right. Yeah.
The,
But
yeah.
this paper is really exciting, right? Because like not every day you get like actual data from non-human primates.
Yeah, it's really noteworthy for that reason. Like I think they had mouse data for a while. They of course had observational correlational human data, but this is like one of those moments where you look at it, it's like, oh, it seems like there's actually something that looks a bit like a therapeutic intervention that seemed to have some sort of dramatic effect, at least in like the best animal model that we have, which is NHPs. So I think
Right.
it's exciting. I think it remains to be seen whether this gets into humans and what effects it would have in humans, obviously. But
Yeah.
So it's an interesting example, though, right? Like FOXO3, right? That was an example where they engineered FOXO3 so that it couldn't be phosphorylated, which kept it more active.
Mhm.
That's like a very minor change. When we were discussing like the whole space of synthetic like proteins and factors, you think about it's a very meticulously choreographed process to keep ourselves alive,
Mhm.
right? Maybe not so meticulous in that like we can survive a lot of different things, but it seems unlikely to me that we could find something that is so, like, let's say, like totally outside the norm of what our biology sees, and that would be good for us.
Hmm.
But I guess it's possible.
I mean, all drugs are in that category, right? Like they're all super artificial and they're usually not good for us in
Yeah,
some capacity, but they're like less bad for us than the default state of disease that we're in.
right. I guess like small molecules are typically not things we ever normally see,
Yeah.
but it interacts with some protein in our body, and it has. That's a
Exactly.
fair point.
Yeah. It's interesting though. Yeah.
I think my take on that would be, I guess, like two like concepts of like roughly the same pattern. I am not in the camp that believes that there's like an evolutionary reason for aging. I think it's just like stochastic damage accumulation. And at some point the body's just not no longer able to handle it. So I think it's not evolution hasn't optimized for us to live longer. And usually things that evolution hasn't optimized for are like malleable. Like if you're trying to optimize like an enzyme that has been like evolutionarily optimized to catalyze a certain reaction, like good luck with that. But like if you're trying to do something that evolution has not optimized for, I think there's a lot of wiggle room there. And the same thing for transcription factors, like I guess in our retro OSKM work in general, I think one explanation for why it was comparatively, I don't want to say straightforward because I think a lot of like accumulated experience went into that project, etc. But like we did get results in like a couple of months, which is not the norm in biology. Not the norm for other projects we've done. And I think like transcription factors are like in during development, they have like hundreds of different functions in like various different tissues. So they're not very tuned to do like one thing specifically well. They're kind of like tuned to do 100 things semi well. But with reprogramming, for example, you mostly care about like this one specific functionality, a few hundred still, but like less interactions than what they usually undergo. And you want to specifically optimize for that function. So given that the factor isn't already optimized for that, it should be comparatively simple to tune your factor to do a certain function. So if you're trying to, I don't know, like I guess if there's some like target of aging, like a process, like a hallmark or something that we might want to alleviate, like getting a factor that hasn't been optimized by evolution to do that should actually be very feasible, at least in my calculation.
So it makes sense that there wasn't really evolutionary pressure to solve for aging. However, like I guess there's a question of when the damage from aging starts,
Mhm.
right? Things like atherosclerosis, like that's kind of unique. Like that's a very specific mechanism that maybe we separate that out from like, let's say general effects of aging of like cells losing their identity and generally functioning worse. Like if that has happened a decent amount of the time when people are still like reproducing, there would be some pressure to fix it. And if like simply reactivating a certain transcription factor could rejuvenate the cell, such as like the Yamanaka factors, seems like evolution would have found that, right? Like if there's some amount of damage occurring at age 30,
Yeah.
I don't know,
But
like I'll reproduce more if you
it's good enough,
reprogram.
right?
Huh?
It's good enough, right?
That's true. Yeah, it's doing it a little bit actually. I mean, it's doing damage repair. So
especially
yeah.
in like the I'm blanking on the gametes.
Yeah, it's doing it
Gametes.
there.
Yeah, it's enriching the gametes. We had learned a little bit about that.
Enhanced damage repair in general compared to somatic cells.
Yeah, we were talking with Christopher Bradley about that.
Is it enhanced damage repair or is it just protected from damage?
Both.
I'm not sure actually.
In protected compartments and upregulated damage repair. In iPSCs, you also have upregulated damage repairs. You can see it in culture.
Does that damage repair, is it similar in any way to reprogramming?
I do think reprogramming undergoes a phase of damage repair as part of the reprogramming process. For example, tying it back to FOXO3, it's known to upregulate certain damage repair mechanisms. I think there was a study where they knocked out FOXO3 during reprogramming, and they just weren't able to reprogram the cells anymore. So I do think there's a factor where and I think this plays into like what I mentioned initially. Why does it get harder to reprogram cells as they age? Because they become more damaged, and at some point, I think the cell is just like it gets these factors. I mean, it's like anthropomorphizing the cells here, but like it gets these factors, and it's like, no, I'm just not capable to undergo this like process anymore, so I'm just going to go apoptose. And I think that's why you just see like in aged cells, you try to reprogram them, you mostly see cell death. And it's just because they're not able to repair all that damage anymore to get to the point where they can become an iPSC. So there's a question. I mean, there's this like somewhat conspiratory sort of like question of like how much is reprogramming rejuvenation versus selection?
Oh yeah, that's very interesting.
That's interesting.
Let me. I want to flip this now. Actually, so aging is something evolution did not spend a bunch of time trying to optimize, and so it should be pretty malleable. But damage repair, like the flip side of aging, is something evolution optimized a lot.
It's a trade-off, right? Because it's energy expenditure.
Yeah.
And if you're, I mean, in the human form, if you're living in like a hunter-gatherer society and you don't know when you're going to eat next, do you really want to spend a bunch of energy on like body-wide damage repair? I guess it sort of happens because at that point you're under caloric restriction, which actually upregulates a bunch of maintenance and repair pathways. But like I guess so there's this, I guess it's like a certain equilibrium where like you do enough DNA damage repair to get through those like times of caloric restriction. But I don't know. I at least part of like I guess you would think that nowadays when you consume for breakfast the amount of calories that a hunter-gatherer would maybe consume in like a month or something, like there should be a way that we should be able to overactivate like damage repair or something to even go beyond what is naturally possible.
And that's interesting because you're not even necessarily saying we need to optimize damage repair per se. Like we just have to turn it up.
I think this is also like I'm reminded of the term antagonistic pleiotropy, right?
Yeah.
This idea that the things that evolution optimized for through the course of our evolutionary history, which is essentially like, can you survive long enough to get to the age of reproduction and consistently like repeat that cycle over the course of many generations? Like, I think optimizing for DNA repair well into your 50s or 60s or 70s probably just got selected against in favor of things that would allow us to get to 25 and through reproductive age, right? And so it's like, it's not that evolution has programmed itself in order for us to, age and accumulate damage and die by age 80. It's more that it really wanted to select for having reproductive fitness until age 25.
Right.
And there was a bunch of like negative stuff that emerged from that, right?
Yeah.
So
do you think abstinence increases lifespan?
Interestingly, yes. It's there are studies of selectively breeding animals, both flies, and I think they've done it in mice as well. And if you delay reproduction, you enhance lifespan. And this is also so there's that, and then when animals live in like more hostile environments, where essentially either you reproduce early or you don't have offspring because your environment is so hostile, you're going to be eaten by like 3 months old, which is basically mice, right? You were essentially forced to shift reproduction to the very beginning of your life, and that decreases lifespan. But then when you do the opposite and you like in culture, you selectively breed flies to just like reproduce later. Like every generation, you just take the flies that reproduce later and you use those for the next generation and next generation. I think the name of the author is Michael Rose or Rosen. They've done these studies in mice and in flies, and I think they were able to extend fly lifespan by quite a significant amount via that strategy. So I do. I mean, it's I guess not in our lifetimes, but like I guess in the long term, if like all our therapeutic approaches fail and we just want to increase human lifespan, like maybe already the late reproduction that we're experiencing now is like gonna extend lifespan over time.
Oh, that's interesting, right? Because
It's already
puberty
happening.
puberty is getting delayed. Wait, no, isn't puberty happening earlier?
It's happening earlier now.
But people generally reproduce like much later.
Oh, you're
Especially with
actually,
IVF,
yeah.
etc. Yeah.
Yeah, that's true.
Well, I guess IVF is no longer in that.
Yeah, but there is birth
regime.
control. So do people physiologically, does the body know that it hasn't actually reproduced?
I think if you're a
Yeah.
woman, yes. I think if you're a man, it's
Yes.
a little harder to tell, but it's like external. But
But I guess even the
I think we can do better with therapeutics though.
Yeah.
Like, we don't want to have to go to that strategy.
I mean, this all does seem similar to the classic like nutrient sensing pathway, right?
Right.
Which is in general, if there's like famine or maybe lack of reproductive opportunity,
Mhm.
like the incentive to live longer is there, and so you're gonna like preserve resources until you can reproduce.
Right.
Those are things that haven't been as effective in non-human primates, right? Like the calorie restriction is like less of an impact than in like mice.
Questionable, I think. I think the studies that were run back then are controversial, and I don't think you can conclude either from the data. I don't think it has been reproduced after that, because there was like the study from like the NIA and like Wisconsin in like macaques, I believe, and one study I guess showed that there was some effect on health span at least, and then the other one showed no effect, and because of that contradiction, people just generally assume that there is no effect. But I do think there are some caveats to the study, as like the food intake of the animals, like the quality of the diet, etc. But okay, generally I would agree with you that like right now we don't have any evidence that that mechanism works at all as well in humans as it does in mice, humans or NHPs. So we probably need something else.
Do you like the who's that guy, like the health influencer from the 80s that people really like? The metabolism guy. Oh my god, I'm forgetting his name.
I don't remember.
I
was
Pete. Peating.
Oh, Ray Pete.
Yeah, Ray Pete. Do you think
I did not know Ray Pete.
Okay. But one of his, so I'm not
Carrot
that
salad.
familiar with him, but there are some people in the longevity space that really like him. He's not very commonly liked, but there are some people who really like him. But one of his things is he thinks like all of health is like metabolism. And I have been really intrigued by this idea, and I want to sort of pay attention to it, which is like there is this idea like we're conserving resources, right? Like if the body knew like it has the resources, like go do more repair, right? Like in theory, like maybe we could live longer.
Right.
Like, are you intrigued? I guess in general, like your thoughts on like the importance of metabolism. Like, does like a healthier metabolism, like you mean a longer lifespan and like more damage repair?
I mean, I am in the camp of people who believe that I do think the GLP-1s are probably going to show increased health span and potentially lifespan for all people, not just metabolically unhealthy people. Because in a way, it is like caloric restriction, right? Like, and possibly a bunch more than that. Also, another example of protein engineering.
That's right. They added a cholesterol group to it and swapped out an amino acid. Yeah.
3 amino acid
Three amino
changes
acids,
and
okay.
fatty acid chain.
Yeah.
23 years of protein engineering to get to a semaglutide.
Yeah, and when it tells you protein engineering is easy, just like 23 years to get to one drug. It's
That's
a
how
lot.
we need models.
It's a lot of work.
Yeah.
Maybe taking things back into like AI for a bit, like, well, there's a lot of talk about how AI is going to, like reach this inflection point of artificial general intelligence and eventually artificial superintelligence, and that will effectively solve aging, and that we just need to get to that point in order to reach the utopia of like longevity medicine. What's the sentiment that you have and that you've kind of gathered from other scientists at Retro, other companies? Like, do you believe that's like a commonly held sentiment, or how do you feel about it?
I do think in bio, I very much believe that there's a possibility it could happen, but you still, even if the system was superhuman at coming up with, like, I guess if you look at like with the best human scientists, like the top 0.001% of bio researchers, like do they in their sleep dream up molecules or like mechanisms and then go in the lab and like just test them and it just works?
The benzene
Like
guy.
The benzene, the PCR guy, right?
right. So potentially that exists. So but you still end up having to test things in the lab to make sure they work. And maybe there's going to be some iteration. So I do think I mean, for what it's worth, I think the model is already superhuman in biology because like biology is just like it's not like years of training in like a very deep domain like physics and only then after like 20 years of training you can actually do any meaningful research. I think biology is like epistemologically extremely flat, right? And like humans, how many genes do you have in your head right now? Maybe like a couple hundred at most. Like a model would just easily memorize all the genes and all their sequences and all their annotations, etc. So I think they're already superhuman at that task. I think the bottleneck is like to actually getting to something like longevity escape velocity is to putting a model into an environment where I can actually just test hypotheses at scale.
Yeah.
And I think that will make things very interesting.
How far away are we from actually testing things at scale? And what do we need to do
Depends
to
on the
get there?
domain.
Yeah.
Um,
That's fair.
I mean, like, I don't know. I mean, maybe the adaptive thing is a good example, right? Like, you have a cell-free expression of your protein binders of interest. So that's like 7 days to get twist. Well, I guess like 4 to 7 days to get twist to print a bunch of DNA sequences to you. 1 day to express them in a cell-free manner. Throw them against your target of interest. So that's like a feedback loop of like less than a week. Or roughly a week, which is pretty good. I think that's like maybe amount like almost the best you can get in bio. But that's like cell-free, like protein binding, then like in vitro, in vivo, like actual like functional animal studies, etc. All that like progressively takes longer. So I think like having some sort of like good proxy assays that have like a reasonable correlation with like the actual thing and ideally like efficacy in humans, I think that will be the bottleneck. But I do definitely in our lifetimes, um potentially I think we'll see like great progress in like definitely in the next 10 years.
What are the proxy models that you view most favorably right now?
For aging specifically or in general?
Aging specifically or in general, either.
mm, I'm not a huge fan of like the clocks.
I don't think
uh,
we've met a single
Yeah.
person who likes the clocks yet.
That's a theme.
um there's so many caveats there, but I do think there's okay, like in model organisms, people do these like resilience studies, and this hasn't really there's not as much like evidence that this replicates in higher level organisms as well. But like, if you want to screen for longevity drugs in a worm, I mean, yeah. Okay, in a worm, there's many ways to go about it. You can literally mutate the whole genome and you can just screen like that. But if you want to like have a quick readout other than lifespan, which is, I don't know, like 30, 40, 50 days, you could just like stress the worm with like, I don't know, like an oxidative stress molecule, chemotherapy or something, and then just measure their resistance to stress. And there's a correlation of that with lifespan in model organisms. So potentially something like that would map onto larger model organisms as well, and that would be faster readouts. Like then you're no longer you have to wait like a year and a half or two until your mice die. You can maybe run an assay in like a couple weeks.
So, like my increasing view on aging, and this has come from like these conversations we've had over the course of the podcast, is that it there probably aren't that many central mechanisms of aging that you can view as a sledgehammer that you can go and re-engineer and bring
Agreed.
things back to a baseline. I think if there's any, partial reprogramming seems to be the most promising bet, but
Mhm.
likely aging is the accumulation of many different mostly independent forms of damage
Right.
that also interplay with each other and feedback on each other, right?
Yeah.
And so I think where my mind goes to of like what that proxy system looks like is you have to have a proxy system that is standardized and very reflective of actual clinical human biology, but also you can measure on a time scale and on a cost scale and a throughput scale that is engineering. It's unlocked by increased engineering, right? It's like basically like, for example, for Moore's law, you can pack more transistors into a chip over time. It's like similar here. Like, can you pack more organoid models or humanized mouse models into a cage or into a dish over time and measure like very minute changes in the course of like a day that
But
Yeah.
it kind of is, right? Like, if you want to look at like what is aging in a human, like how do you measure aging in a human? I guess like one measure is like, you put them through like a party night at New Year's. 20-year-old person gets back up the next day, doing totally fine. 50-year-old person, be down for a couple of days and will not want to drink in like a very long time. So like the recovery after a stressor, like I guess in this case high alcohol exposure and a late night of partying, is like the like bounce back time from that, like very, very different from an old person to a young person. Hospital stays, you break your leg or like I guess your hip. Like as an aged person, you might not at all recover from it, or you will stay in a hospital for a very long time compared to a young person. So I think just like like resilience, like the ability to bounce back from stress, like I'm not implying that it would be a good thing to screen against, but it's definitely a biomarker of aging that could be very interesting.
That to me is like a really important grand question, right? This idea of what are the biomarkers of aging that are measurable at a clinical scale, but
Mhm.
also measurable at an in vitro or animal model scale,
Yes.
and that
Hmm.
are mapped between the two in a way that allows you to transfer a lot of what previously was limited to very expensive, very long human studies, and you can now run those studies in a dish or in an animal model, right?
It's such an important question because like I feel like when you I feel like when you look at the molecular level or like for clocks or like proteome or any of the omics measurements, I feel like you're really pigeonholing yourself because like I feel like with age everything breaks down everywhere all at once, right? So like you're going to find a ton of things that will go down or up with age. It doesn't mean that any of these things are meaningful and or targetable, right? So yeah, very I mean, I think literally the however you want to value an aging drug, like the $1 billion question.
So it makes me think about, cells in a dish don't really age, right? Like you're not really seeing the phenomenon.
Right.
But I wonder what is the cell in a dish equivalent of, a late night of partying and alcohol? Like I wonder, what if you took
You
a
just add
biopsy?
alcohol to the dish.
I was just thinking that, but how do you make them party?
Oh yeah. Gotta shake them a little bit.
I wonder if you had a good stress test for cells. I'm sure there's something that exists, and you took some cells from an older person, like some easily accessible cells from an older person versus a younger person. Could you, yeah, like, then give them stress tests or whatever, and like start gathering some interesting data that way?
Interestingly, like even the dissociation of cells from an old person and like digesting them and having them attach, like going from this snuggle, warm environment to like attaching them to a plastic plate is already a stress test.
Mm. The
And
adherence itself is like a non-trivial problem for
Yeah.
cells to manage.
like much less efficient in age cells versus young cells.
So I maybe one point that comes to mind here is I do think there are some really interesting examples of these like really interesting preclinical models that seem to map some important biology in like clinical physiology. So there's this work from Ann Carpenter that recently got published just a couple of days ago, and essentially they were taking skin fibroblasts from normal patients and patients with major depressive disorder and patients with
Mhm.
schizophrenia, and they were looking at different presentations of mitochondrial organization in the
Mm,
schizophrenic versus normal patients and the depressive patients. They found pretty consistently that mitochondria in the schizophrenic patients actually cluster much closer to the nucleus and even
interesting.
from skin cells. And so there's this sense that there's some sort of like general mitochondria related, metabolic related, energy like bioenergy related disorder that is kind of underlying all schizophrenia. And the opposite happened in major depressive disorder where the mitochondria clustered further away from the nucleus. And so
Hmm.
it's like, can you start to view the in vitro model of primary fibroblasts as like a way of determining like whether or not it's probably not diagnostic, but whether or not a drug is influencing mitochondrial clustering and whether that's actually related to, like some sort of mechanism of action that's related to like disease physiology, right? And like disease modification.
Right, right, yeah. Um, I mean, it goes back to the point like there's like tons of things that break down with age and figuring out which ones of those would potentially be causal. I guess it's the same for disease, I guess. Like you will see a lot of things that potentially change and then figuring out which of them are causal so you can actually use them as a model. Very interesting question. I mean I'm really curious about like organoids and all of that in general. I mean even what you're talking about like stressing cells in vitro, like maybe a meaningful readout, but then again, like the data is mostly for model organisms, and there you like stress the whole worm. So like even an organoid should be more relevant as like for stressing an organoid in culture than just even stressing a bunch of individual detached cells. Oh, there was a really cool Oh, there was a really cool paper, another part of protein engineering. There was like the naked mole rat.
Yes, we talked about this paper the other day, actually. C-gas was the
Yes.
enzyme name, right? And so, like, in the naked mole rat, there's this enzyme. Oh, it's in all mammals, I think, but C-gas is essentially an enzyme that's involved in homologous recombination mediated DNA repair. Apparently, the naked mole rat, which is this mouse that lives like, or this rat that lives like 37, 38 years or something, there's like no biological aging in these rats. And so they live like an order of magnitude or something longer than normal rats. And it has these 4 amino acid differences between other like the mole rat, C-gas, and other rats and other like other mammals. And so they like basically implemented these 4 amino acid changes into I think a normal mouse, and they saw crazy stuff like, the hair started to get degrade and they looked younger, and
Wow.
it's crazy.
So is that the answer?
I think we solved
Podcast
it.
over.
I think we solved it. I think it's it. Yeah. No, obviously we got to do some more stuff, but like I would be really interested to see that happen, like the same study happen in NHPs. And I'm potentially ready to put a little like mole rat C-gas in my body. Yeah, it's very interesting.
It's really interesting that evolution, in one case, it seems like evolution might have solved aging. Even like I wonder if the naked mole rat, like its predecessor, whatever species, like had a lot of evolutionary pressure to figure out the aging thing for some reason.
Yeah, I think the paper proposed some explanations for this, and I'm not going to pretend to have remembered it. I don't remember what it proposed. It has something to do with the fact that the mole rat, because a lot of the species that end up having unnaturally long lifespans end up being either underground or
Yeah.
deep in the ocean. So there's something about being like separated from light and a lot of other evolutionary pressures, because you're like relatively isolated as an organism, that seems to allow for kind of a different mode of evolution, like a different manifold of evolutionary pressures, right? That you're then traversing. And I'm just using words. It's interesting though. Yeah.
All right.
Maybe we already know the key to.
Yeah, I mean, I'm gonna go
radical
inject
lifespan
that
extension.
immediately.
I'm gonna get the FOXO3. I'm gonna get the C-gas.
If anyone wants to sell to me C-gas or FOXO3 that I can put into my body, reach out. Paying top dollar for that. But it's going to be a subscription pricing, so you have an incentive to keep me alive. I don't want to die during the first treatment. Thank you, Rico. Where can people learn more about you?
I don't really use social media these days. If anything, like Twitter.
Great.
All right. And we'll put your info in the description. Rico, thank you so much for joining us.
Thank you guys.